Okay. Uh, okay. Can you hear me, Christine?
Yes. Sorry, I don't know why it wasn't working.
Oh, it's okay. It's okay. Okay. So, good afternoon, everyone. We're back to the event, and with a great pleasure, I would like to call Professor Christine Meynard to talk to us right now. Let me just talk a little bit about Christine. She started her career in Chile, and then she pursued her PhD at the University of California. After receiving her PhD, Christine has done two postdocs, the first one in her home country, Chile, and the second one in France. Then she got a position at the French Agronomical Research Institute for a few years. After that, she moved to the United States as a faculty at the Virginia University of Marine Science. Finally, she came back to France, where she works up to now as a researcher at the French National Institute for Agriculture, Food and Environment. Her research is framed within the disciplines of macroecology, biogeography, conservation, and community ecology. She is a theoretical researcher studying analytical methods and its links to theoretical ecology. Her studies encompass different taxa from different places around the world. Also, she works on potential applications of theories in real-world issues, especially those related to agriculture and climate change. So, Christina, whenever you're ready to start and
Okay.
share your screen, we are here for you. Thank you very much.
Okay. Thank you. Can you hear me there? Is that
Yeah,
working?
it is.
Okay. So I'm gonna start with the screen sharing. There you go. Okay. Well, thank you for that introduction, and thank you for inviting me to participate in this workshop. It's very exciting to be among the speakers of this event, and it's also very exciting for me to be talking to Latin American students and researchers. And so today I'm going to try to give my vision of metacommunities and how we can link empirical data to theory, or theory to empirical data. And I wanted to start off by just highlighting what the interest, the importance of this is. So in the early 90s and late 80s, there was some talk about the importance of scale in ecology. And Simon Levine, in a very interesting paper, highlighted some of the main, the importance of scale in the context that we're living today. That is a context of biodiversity loss and globalization. And we are at a moment where we really need not only to understand, but also to predict. And so in this context, he argued that we need to get out of the little boxes of local studies. We need to find something that allows us to really answer to the challenge of understanding and predicting the effects of humans on biodiversity and how to prevent further loss of biodiversity. And so elements of that involve interfacing, understanding the interface between phenomena that occur at different scales, but also understanding what are the mechanisms that are behind the observed patterns. And behind all of this is the idea that we need to quantify things in local systems that allow us to sort of get at a common ground and compare systems across different communities. And so we need to find some way of finding rules that could be applied to different systems, because without that, we will not be able to understand the effects of global disturbances, but also predict and try to anticipate how to prevent the loss of biodiversity at large scales. So in that context, metacommunities emerged as an idea a bit later, but there were a bunch of different ideas and topics that were old community ecology traditional questions. Some of these were probably mentioned in the talks this morning. There is a bunch of literature on species interactions, a lot of modeling of how those species interactions are play at a theoretical level, and how those could be translated into patterns in real communities. A big question in ecology forever has been, how do species coexist in space and time? So all those different questions have been sort of developing in disconnected ways for a while, even centuries before the metacommunity concept emerged. But in the 80s and 90s, there was something cooking that made it so that things were coming together and people were trying to integrate. We were trying to integrate scales and fields and methods. And so, of course, metacommunities is in part inspired by the concept of metapopulations. So this idea that populations are connected between them to a lesser degree by dispersal, that environments are sometimes patchy, and so there might be areas where populations can survive, but there is a matrix in between where the populations cannot survive. There is also the concept of macroecology that was born in that time period, where we were trying to search for general patterns across communities at large scales. And I think this is a good book that summarizes the state of the art of things before the metacommunity concept was really out there. And so in this book, there is a lot about all these different things, and there is a lot about the role of null models and the importance of null models when we are trying to figure out what is it that is structuring the different communities we're studying. But there is also a lot about scales and functional relationships and all those sort of things. And I think if there was a catalyzer for anything, it was this book published in 2001 by Stephen Hubble, where, so it was the Unified Neutral Theory of Biodiversity and Biogeography, where he started off with a very simple model that really lacked a lot of the mechanisms that most ecologists would recognize as important in communities. And he just said, well, all species are equal. Let's suppose that all the species are equal, and that there is just random drift, and there is competition for space. But other than that, there is very little mechanism in it. And just starting off with this very simple model, he could actually generate a lot of the patterns that people had been studying in macroecology and in ecology in general at different scales. And I think a little bit as a response to that, more and more people, more and more ecologists started reacting to that lack of mechanism. And the classic book on metacommunities was published in 2005. and it proposed different mechanisms to explain this different ecological patterns. Now, let's look at what the unified neutral theory really gave us that I think was fundamental to develop the metacommunity theories after that. First of all, he was trying to integrate across scales, both spatial and temporal. And that's something that metacommunity theory aims to do. We were trying to incorporate evolutionary scales, so incorporating the effects of mutation and speciation into the communities. He was also mixing in all sorts of theories, from ecology to macroecology to evolutionary theories. And so there was really an effect of, okay, we really need to integrate different scales, different theories, but we need to do it in the context of community ecology. So that was like the positive trigger, I think, that the unified neutral theory did for ecologists at the time. But at the same time, you know, a lot of ecologists were frustrated that a theory that could incorporate so little mechanism could be used to explain all these different patterns. And so that made it so that it was important to actually incorporate all the mechanisms that community ecology had been documenting before into something more structured, something that could be unified, but that recognizes that species are in fact different, that it is important to contrast, the contrast with the null hypothesis is important, but it's not the only possible answer to the problems. And so, and the other thing that the neutral theory made for us was to make it very clear that there is more than one mechanism that can generate the same observed pattern. So with that in mind, the concept of metacommunities was born as this extension of the metapopulation concept, where we have somewhat isolated communities that are connected between them through dispersal, but through limited dispersal. And as a whole, they form a metacommunity. But it is also important to notice that in some of the theoretical models, the conceptualization of that was resembled more to this graphic, which is there is a regional species pool, and all the evolutionary mechanisms and the interesting sort of large scale things are happening in the species pool. But this species pool is interacting with this local community. So these two scales are somewhat disconnected. So even though the concept is there, sometimes it's a bit of a challenge of how do we actually conceptualize the problem of scales, both spatial and temporal, and what the effect of that is in both in the theory and in the practice. So in the initial metacommunity theory, at least the one that was presented in the book in 2005, there were 4 different paradigms that were distinguished. The neutral, which corresponds more or less to Hubble's theory, where all the species are equal, but there is random dispersal, there is competition, and therefore there is drift and other processes that can generate the observed patterns. Then there is species sorting that corresponds to environmental filtering, acting on species to filter out the species that are not locally adapted to the environment. Patch dynamics, which deals more with the colonization extinction dynamics. So it rescues more the aspect of patchiness and how dispersal and competition can be in a trade-off, where if you're a poor competitor, you have to be a good disperser in order to survive in the landscape. And finally, the mass effect, which was all about both having an environmental gradient or patches of different environmental quality, where the species could survive better or worse, depending on where it was in that gradient. But there was spillover, dispersal spillover, that allowed for some species to survive where they should not be without or in the absence of dispersal. So those for a long time have been the four main paradigms in metacommunities. However, more recently, there has been more and more a recognition that the paradigms are overlapping between them. And what is really behind and what we should really be looking at are the processes, the fundamental processes that are behind. And those processes are usually dispersal, environmental filtering, species interactions. And then there is a sort of two different views, whether you consider stochasticity to be a fourth different process, or as I tend to think about stochasticity, more about something that is inherent to every ecological process. So stochasticity is everywhere. And then the role of evolution depends on the scale at which you're looking at, but it can affect all the processes at all the different scales. And so the question now is, okay, we have this sort of rough understanding of what the important processes are. How do we distinguish these different processes in real communities and in real metacommunities? Studies dealing with metacommunities have been looking at questions related to temporal and spatial scales, but also, are all these mechanisms the same or similar in different types of environment? For example, do tropical metacommunities have the same sort of community assembly rules than the temperate ones, etc.? And then what's the role of evolutionary versus ecological processes between all of this? Now, the reason why I started with the theory is that theory and practice are linked. If we don't have a clear picture of what we have as a tenet for our ecological expectations, then it's going to be even more difficult to understand our empirical data. And so as theory evolves, the tests that we develop to test those theories should also be evolving. And one key aspect that is often neglected is the role of simulations in this, because sometimes we may have a very clear conceptual picture of what one mechanism or one process should result in, but then when we put it into practice, either through a theoretical model or through simulations, we realize that our expectations were not either fully realistic or fully complete. So that feedback loop, I think, between theory, practice, and simulation is something that we should keep in mind more often in ecology. Now, for the rest of the talk, I'm going to be focusing on linking empirical data and theory. There is a whole set of studies that try to do this through experimental work, usually using microcosms, organisms that reproduce very quickly and that you can manipulate at very defined small scales. This is not at all the focus of my research or this talk, so I'm not going to be talking anymore about that, but I just wanted to mention that this whole field has also contributed a lot to the development of the metacommunity theory. And my focus is going to be more on the observational side of things and how can we use statistics to actually try to understand what are the different processes acting behind the composition of natural communities. And so the question is more about how to distinguish the different processes in real metacommunities. Now I'm going to distinguish between three types of strategies. The first one involves using spatial patterns of diversity. And so I'm going to talk a little bit about that. Then there is using phylogenetic and functional patterns. And the last one is using simulations to try to know, understand what kind of measures of diversity or what kind of analysis can be helpful in different situations. So the first strategy is using spatial patterns. And before I start looking at that, I wanted to clarify two different concepts that that are essential to understanding spatial patterns. The first is autocorrelation. And autocorrelation usually deals with more local patterns where the communities that are closer together or the environments that are closer together are more similar to each other than those that are distant. And so this is more of a relatively fine scale pattern. And in order to analyze that sort of spatial pattern, you need to know geographic distance and compositional distance between communities. The second one is spatial trends. Spatial trends are more of a large scale pattern, and they usually deal with climatic and environmental data that are co-varying in space at the same time. A classic example is if you move to higher elevation sites, or if you go towards the equator, you're going to find that a lot of different variables, temperature, precipitation, and humidity, and a lot of different things are varying at the same time. And so that's a spatial trend. So this is sort of a context for what's coming next, which is how do we use that sort of pattern to try to deduce the processes that are behind community assembly. So in the first line up here, you have different colors representing different species. If we start with a community of 3 species here, and the red one has a lower dispersal ability than the black one, in all cases, you're going to find spatial autocorrelation between the composition of the communities, right? The community that is here is more similar to this one than it is to that one there. But also notice that this dispersal limitation means that this spatial autocorrelation is not related to the environment that is varying or not varying in this line, okay? In the second line here, we have no dispersal limitation. All the species disperse the same. And whether or not the species is here and not there is more of a function of random colonization and extinction events, but it's not related to the dispersal ability of the species. And in the last one here, we have an example where we're going to suppose that there is an environmental gradient varying along this line. And the three species have the same dispersal ability. They can move 3 sites along, but the red one prefers the environment on the right, and the green one prefers the environment on the left. So the whole principle of trying to use spatial correlation and trend in this has to do with this relationship. In here, we would expect that the trend would be significant, but that spatial correlation is 100% linked to the species and not to the environment. Versus here, we won't find any environmental relationship as long as we control for the autocorrelation aspect of it. So there are different ways of trying to tease apart the roles of environment and autocorrelation. But they often assume that autocorrelation has to do with dispersal and environmental trend has to do with environmental filtering. So this was part of my thesis project, actually, where I was sampling bird and vegetation communities along the parts in Chile. Now, the advantage of Chile is that it is a very long country, and there is a mountain range on the coastal line here, and there is, of course, the Andes, which is another mountain range on the other side. And so you have very steep environmental gradients from west to east and from north to south. And that makes it so that the spatial trend and the autocorrelation can be somewhat separated. And so by looking at turnover, so the change in composition between the different communities, I could see that the species were organized along the environmental gradients, and that was independent of the spatial trend of the climatic variables. And the environmental effect was actually much better at explaining the composition of the bird communities in these places than the spatial trend and the spatial autocorrelation per se. So that suggested that the environmental filtering was playing a strong role over here. But if I remained stuck on regressions only, I could see that there was a strong effect of vegetation and a somewhat smaller effect on climate, and I wanted to go a step over that. And so I used path analysis to try to look at the direct and indirect effects of these variables on bird diversity. And what the results showed was that the vegetation structure represented by this NMDS axis here had a direct effect on bird diversity, whereas the climatic variables had an indirect effect that was significant over the vegetation structure, but not so much over diversity per se. So the climate did have an effect, but it was mediated through the effects on vegetation structure. And similarly, fragmentation, so the area of the fragments in this case, had a strong effect on vegetation structure, and it did have a direct effect on diversity, but that was smaller than the effect it had on vegetation structure. So that's an example of how you can combine different types of analyses along with the analysis of spatial structure to try to tease out the role of environmental filtering on bird communities. A more common practice is to use variance partitioning. And variance partitioning is a multivariate strategy where you have a response variable, which is usually a matrix of either presence, absence, or relative abundances for a set of communities. And you're going to be trying to explain the total variance explained. You're going to be trying to divide it into what's the part the environment can explain, what's the part of dispersal, and what's the interaction between these two other parts. Now, this has been on the books for much longer than the metacommunity theory, because it can also be used to tease to separate the roles of different variables in any type of response variable. And if you're interested in this type of analysis, I think you should visit the classic numerical ecology book by Legendre and Legendre, for which there is both a more recent version of it, and there is also an applied version using R, which is also very useful, of course. And so in 2005, Carl Codney published a paper that is, I think, a classic on metacommunities, where he did a meta-analysis trying to gather as much data as possible on communities across the globe. And one of the results that I think is very recurrent when you use variance partitioning is that, first of all, one of the main components, or one of the components that remains very large overall, is the portion of the variance that we cannot explain. So you do all this analysis, and at the end, you end up explaining something between 20–30% of the variance on your system. And the second aspect of it is that the part explained by the combined effect of the environment and space is often more important than space per se or environment per se. And I asked the question to Michelle this morning, because the interpretation of what is this interaction, what is this component that is composed of both the environment and space, is not obvious from the biological point of view. But statistically, it means that the structure of the environmental variables is so similar to the structure of space, or it has such a strong spatial structure, that we cannot actually separate what would be attributed to dispersal versus what would be attributed to the environment. In my own work, I have used variance partitioning quite a bit. Here on the left, there is these are bird communities in agricultural landscapes, and Laura Henkel, who was a postdoc, still a postdoc, studying agricultural landscape, did this analysis where she was varying the scale, but notice it goes from 200 meters resolution to 1.4 kilometers resolution. And the total variance explained actually decreases as we increase the environmental gain. But in this case, the environment explains a large portion, or the largest portion of the variance. Still, in most cases, we never explain more than 20% of the variance in the system. And on the right, it's similar analysis that I did with the plants in alpine grasslands. In this case, the scale is, we go a step higher, because we start at 1 kilometer resolution and we end at 30 kilometer resolution. And in this case, the variance explain increases with the resolution, but the spatial structure, the component of spatial structure increases with resolution. So variance partitioning is very widely used in trying to understand the role of environmental filtering in metacommunities. However, a number of issues arise when we're using it. The first one is, how do we represent space? And there are different ways of doing this, and from the simplest inclusion of XY coordinates in a polynomial regression to the more complex eigenvector maps, PCNM, and multivariate methods that try to include as much of both the trend and the autocorrelation in the spatial structure. The same can be said about how do we represent environmental gradients. We can either take raw variables, if we have a good understanding of what the important environmental gradients are in our communities, but most often we tend to collapse a bunch of different environmental gradients into PCA or other sorts of ordination analysis, so that we can forget about the correlation between them and try to understand the combined effect of different gradients in our response variable. Another level of difficulties come when you try to understand what are the best measures to represent differentiation between communities. This is a whole different topic that I'm not gonna be developing, but not all measures of turnover are equal. And so when we are trying to measure turnover and then deduce processes from there, we need to understand the properties of the indices that we're using. And then there is a question that I mentioned before: what is really the biological meaning? Is there a biological meaning really to the environment-space interaction? Or is it just a statistical artifact because we cannot really separate space and environment? And then in variance partitioning, the role of ecological interactions are really absent. Unless it goes through environmental filtering, there is no way of using variance partitioning in this context. Now, all these techniques can be modified and extended to do different things. This is just an example of Sylvie Clapp, who tried to have she was a student with Stéphane Rey. They modified the classic variance partitioning to try to incorporate the effects, or try to correct for the effect of a neutral dynamic behind the observed patterns. Another way to go is to use co-occurrence analysis. So if we go back to the classic community ecology literature, there is a bunch of papers showing that if there is competition, species that are competing between them, they should exclude each other. And a lot of the debates in the 80s about the importance of null models in ecology had to do with this, and with how do you define a null model to determine the significance of the co-occurrence patterns. Because if you make it totally random, you're not incorporating enough biology to it. But if you make it if you incorporate too much biology, then you're doing it a mechanistic and non-null model. So conceptually, it's an important idea that some species will co-occur less often than expected by chance, but the challenge then becomes what do we define as expected by chance? On the opposite, if we are expecting environmental filtering, then the expectation is a bit the opposite. You know, species that are similar and species that that are adapted to the same kinds of environments will co-occur more often than expected by chance. And so in this topic, in general, the books and papers by Nicolas Gotelli are excellent to try to understand where the debate lies in this area. The other issue is how do you define the species pool? Because if you have a set of very close communities versus a set of of communities that are in a very wide biogeographic area, it makes a difference in terms of what the species pool should be. And the definition of the species pool is once again going to influence what you define as the expected by chance. So again, there's this tension between extent and testing, but also extent and mechanism that I think Michelle was mentioning a little bit in the presentation this morning. Presley proposed this meta-community framework. I think the name is unfortunate because it's a framework based on co-occurrence that is not fully overlapping with the meta-community theories and paradigms that have been proposed. But it's very interesting and very useful because he was trying to integrate the multivariate types of analysis we talked before with co-occurrence analysis that can deal with competition. And he proposed that we could use 3 stages, 3 different types of analysis that he called coherence, turnover, and clamping. And he proposed this sort of decision tree to try to classify the meta-communities into different types. And so the first type of analysis, coherence, have to do with the level to which different species are responding to the same environmental gradients. And to do that, you use ordering of the species in terms of their occurrence along environmental gradients. And so some sort of ordination technique and ordering of the species by environment or species by site matrix. The species range turnover has to do with how often the species ranges replace each other and whether they are replacing each other along a gradient or not. And the boundary clamping is looking at whether or not several species are changing at the same time. So how aggregated are the range turnover between the species? So some examples taken from that paper just to show what it means. So if you order the sites by species matrix from the sites that have the most species to those that have the least species, and the species that are more common to the species that are less common. Of course, this is an ideal situation that you will never see in reality. But this is a community that is perfectly ordered versus one that is also perfectly ordered, but where the species are clumped. They are turning over in a perfectly ordered groups. So they are doing it all together by groups. Versus in the middle, they are acting a little bit individualistically. They are randomly changing along that gradient, but they are, in the three cases, they are responding to some sort of environmental gradient that makes it that some sites are richer than others, and it's quite perfect in this case. Another example from the same paper, where you can distinguish Clementian versus Glisonian structures. In ordering the communities, he distinguishes a case where the species are changing in groups. So there are groups of species that are responding to the same threshold in this environment. And so that's what we call the Clementian structure. In the Glisonian structure, they are changing across this environmental gradient, but they are doing it in an individualistic way. And this one here in the middle is perfectly nested, meaning that the richest communities or the poorest communities are a subset of the species in the richest one, but it doesn't correspond to either the Clementian or the Glisonian structures. So that, for me, was a very interesting conceptualization of how we can try to link and use the analysis tools that we have with real communities. And I applied both variance partitioning and the Metacommunity framework to the alpine communities I was talking about before. And the logic here is shown in this table here, because variance partitioning cannot look at whether or not the assembly of the communities is random. It cannot look at the role of competition, but it does a good job at looking at the roles of environmental filtering and dispersal, at least in theory. Versus the Metacommunity framework, which does not allow for looking at dispersal, but it does allow for looking at environmental filtering, competition, and random assembly. So using both strategies at the same time, I hope would have given me more of a clear picture of where we stand. And so the idea was to use the analysis at different scales to see whether or not the role of environmental filtering was changing and competition was changing across space. And I was expecting that I would have detected competition at the finest resolution, and that environmental filtering was going to be more important at larger resolutions. But in fact, I did not find any evidence of competitive exclusion at any scale. There was strong evidence for a Clementian species turnover, so there were several groups of species that were changing along that gradient in bunches, in groups. And there was strong evidence for environmental filtering and dispersal. And there was evidence also of an increasing role of dispersal across coarser resolutions. We can also try to look at phylogenetic and functional patterns to try to detect different processes in the assembly of communities. So if we think about environmental filtering in a mountain, in this example here on the upper right, we would expect that species that are low in the mountain are adapted to warmer climates than species high on the mountain, for example. So in terms of functional structure, we would expect these species to be similar between them and similar between them within the communities, but to be different between the different communities. And if we also make an assumption, which is a strong assumption, of phylogenetic conservatism, meaning that the species that are phylogenetically closer together are also more similar functionally, then we can also test the same type of ideas on phylogenetic structure. So phylogenetic clustering within and phylogenetic over dispersion between. And then you can ask what the role of evolution on all this is. And so you would expect that similar environments, for example, into different continents, would have some functional convergence. You know, similar species inhabit deserts all over the world. So some functional clustering, despite the fact that they could be phylogenetically very distant. So you can start playing with the relationships between phylogeny and function, and within biogeographic regions, between biogeographic regions, to try to look at the interplay between evolution and competition and environmental filtering. If we simplify all of this, of course, there is a lot more complex than this, but in short, if you look at the functional structure, you would expect that competitive exclusion would result in functional over-dispersion. Environmental filtering will result in functional clustering. If there is evolution with environmental filtering, and you're looking at different continents, for example, you would find, or you would expect, convergence of traits in similar, even though distant, environments. But if we expect, if we have competitive exclusion, even with evolution, we would expect in the long term to produce some differentiation of locally coexisting species. There is a whole debate about the usefulness of phylogenetic structure in this context, but in general, if you are using phylogenetic structure within this context, you need to check whether or not there is phylogenetic trait conservatism before you can actually say something about the potential role of competition and environmental filtering into the structuring of communities. A while ago, we did a workshop kind of similar to this, but in life. We came together and reflected on how useful phylogenetics could be in enlightening metacommunity theory, and so this paper reviews all the different types of things you can do with it in this context. But the use of phylogenetic and functional diversity in biogeography and in metacommunity analysis is kind of recent, and so here we were just trying to check whether or not we could take phylogenetic and functional diversity indices and relate them to classic biogeographic theories and relationships. So in the first line here, you have taxonomic diversity for birds in France, and there is a increasing relationship between local diversity and regional diversity. And you can find the same type of relationships with phylogenetic and functional local versus regional diversity, but they are not exactly the same. They are similar, but sometimes they reveal different things than the taxonomic facet. Same thing looking at variance partitioning. when we look at the role of space and environment in terms of taxonomic diversity, space comes out as the most important variable in this particular study. But when we looked at phylogenetic and functional diversity, it is the interaction between space and environment that comes out as more important. And the same thing can be more or less seen when we look at turnover between sites. And then if we look at the spatial distribution of the other two facets, so phylogenetic and functional diversities, although there is a strong correlation between these two indices and taxonomic diversity, their distribution does have some important differences that are then translated into how we think about conservation in this context. And in this particular case, the black line here representing the level of protection in the current protected network in France. Well, taxonomic diversity is much better represented than the functional and phylogenetic components in this case. Similar result for fish in the Mediterranean. And here on the right, this is a similar graphic, but not exactly the same for anurans in Brazil, a study that I participated with one of the students of the NICE, and where we showed that actually focusing on functional and phylogenetic diversity would have given us a different answer in terms of what the important sites are for conservation purposes. So that's from conservation perspective, but from the meta-community perspective, we would like to know what is the relationship between the species stress and the environment in order to understand, how are species being filtered in these communities, if they are. And this has been named the fourth corner problem. And it is the fourth corner because, the environmental matrix, so this would be a matrix where you have different sites and different environmental variables. This was called the R matrix. The species stress is usually called the Q matrix. Don't ask me why, but this is the names that the first people who published this gave them. And the abundance matrix is called the L matrix. And so they call it the fourth corner problem because we have these three matrices, the environment, the abundance, and the species, which we would like to relate. And so we're looking for the fourth corner, which is this relationship between these three other matrices, but in particular, what are the traits that are affected in different environmental gradients, by different environmental gradients. And the initial method proposed by Doladek involved using analysis where you would look at each trait for each environmental variable, but this could be a very long, exhausting analysis. And so the problem as defined by Bray and Legendre later on, that was called the RLQ method, is actually trying to use multivariate analysis to find this relationship. And so again, the methods evolved and people are trying to implement new things with them, combining them in different ways. And this is one of the extensions that I feel are useful, where first you use the multivariate relationships between these three matrices to try to understand globally what are the main axes, both in terms of traits and environment, that are driving the differences between communities. And once you have that, then you can apply the fourth corner approach, meaning you can analyze trait by trait within the axes that were relevant, so that you can have a better understanding of what is really going on behind it. The problem with these types of analysis is that you need to spend a lot of time interpreting the results, which look a little bit like this. So if you do the multivariate analysis first, you will end up with a cloud of points representing the species and where they are located with respect with these two axes here. And the two axes are themselves a combination of environmental variables or trace. So you have to consider very carefully what all this means. And then using this strategy of combining the two methods, you can take the main axis of the environment or of the trace and test the significance between of each one of the variables that were used within each one of the either environment or trace axis to try to see which ones are significant and in what direction. So in this case, for example, the red is positive relationships, the blue is negative relationships, and the greens are not significant. So in the end, the process has allowed us to identify the main environmental variables, the whether or not the relationships were positive or negative, and also the main life history trace that affected the species composition in the different environmental variables. The holy grail of statistical analysis that I think every statistician would like to see is some sort of combination of analyses that could allow us to look and to separate the effects of environment on species trace, taking into account phylogeny. And in that, there has been another set of papers developing different methods for this. For example, this paper, Braga et al. In Ecology a couple of years ago, they developed or extended the RLQ method, but constraining the permutations that are used to look for significance between the different variables. They constrain it using a phylogeny. Another approach proposed by Pavoine earlier was to first look at the relationship, the links between species trace and phylogeny, then in parallel look at the links between environment and space, and then perform a classic RLQ. So that's another set of approaches. And finally, and very shortly, the potential to use simulations has been, I think, neglected, but is sort of coming out nicely lately with some new publications in this direction. But the idea is that we have data from the real world, and we have some understanding of how that works, and we can actually simulate the different processes to evaluate the statistical tools that we have, and then try to find which is the best strategy to deal with the type of data that we have. And in that spirit, a while ago, I participated in this study where we simulated different types of assembly processes, generated communities in different types of landscapes to try to look at all the different sets of indices, including regional, local turnover indices, and including phylogenetic and functional indices. And we were trying to see whether any of those or any combination of those could help us better understand or identify the processes that were behind community assembly. And the big, the results are in the end very complex because we had a big number of indices behind the scenes. But one of the results that stands out is that there is no single index that makes it all by itself. There's no single index that will identify the correct process behind community assembly. However, the combinations that came closer were combinations of indices of turnover with indices of environmental differentiation, and also indices that included functional diversity. So in the end, the results are kind of complex, but they allow us to sort of hint at the idea that we need to combine different types of analyses and adapt to the situation we're dealing with in order to actually understand what we're doing. So in conclusion, we have seen that there are multiple approaches to try to link theory and real communities. In general, they deal with spatial structure, phylogenetic and functional structure, and co-occurrence. I don't think there is a single strategy that is likely to work all by itself. I think we need to adapt our analysis to the systems we have at hand and the objectives of any particular studies. But in general, I think that the combination of different types of analysis can provide some robustness to the results that we get. And of course, the development of new statistical tools that make this link remain central to metacommunity applications. For me, I'm just starting a project where we are looking at a big data set of more than 500 plots in agricultural field margins across France, where we have plants, coleoptera, birds, and worm data. And I have a student, Isis Ponas, who is going to be looking at the non-intentional consequences of agricultural practices on biodiversity using this metacommunity perspective and all these types of multivariate and spatial analysis. So I'm pretty excited about that. I am very open to collaborating and hearing from students all over the place. So if you want to contact me, here is my email address. You can look at a little bit more about my research on my website. I am a newbie in Twitter, but maybe when I learn how to use it, I will be posting some interesting stuff about metacommunities. And then I would also like to encourage you to look at the events that are programmed for the International Humboldt Day coming up the week of the 14th of September. I will be participating in one event in the context of women in science, and another event for the Journal of Biogeography. And finally, I am one of the deputy editors-in-chief of the Journal of Biogeography. We are always looking for more diversity in our publications. So if you have ideas or just questions, don't hesitate to send us a little inquiry letter and we will look into it. And with that, I can give it up to questions.
Okay. Thank you very much, Christine. Thank you for accepting the invitation. It was very, very important for us. And thank you for the speech. It was a success. We have some questions here. I would like to start with this one. André Padial asked you, what about the residual percentage of variation in variation partitioning analysis? How much do you believe actually represents randomness? And randomness could be interpreted as a lack of theoretical background to explain metacommunities?
The unexplained variance is always an interesting question. Of course, the first obvious thing is maybe we did not include the relevant environmental variables in the analysis. So that could be it. Or we did not include them at the appropriate resolution or extent, or in the good summary of whatever we're representing. So, for example, many times a hypothesis have to do with productivity of the environment, but we rarely actually use productivity. We use the proxy for productivity, or temperature in combination with precipitation, assuming that it will summarize the effects of productivity. So that is one possibility. The second, to me, is not so much randomness, but stochasticity, meaning, anything. You can represent any ecological process with a mean and a variance. So when you say, individuals in a population are adapted to 20 Celsius degrees, yes, but there's going to be some that are adapted to 22 better than in others at 18. And so environmental filtering, dispersal, same thing. You can say on average, individuals disperse 20 kilometers, but there are some that are going to disperse 100 and some that are going to disperse 5. So dispersal also has a mean and a variance. So all the different processes have a mean and a variance. So there is stochasticity within them. And the same, you can say the same about competition. Some individuals, most individuals of one species are going to win over the other species, but maybe there are some that are going to win on the other direction. So there is stochasticity in all the processes, and that, I think, explains some of the randomness. Unfortunately, I don't think we have a way of knowing. There is, of course, models that try to simulate neutral dynamics and try to see whether or not a particular system fits that neutral dynamics. So that's another way to go. And that's why I suggest that it's always better to try to combine one type of analysis with another, because there is no one analysis that is going to allow you to completely solve the issue. So that's the short answer for it.
Okay, let me make the second question. Christine, thanks a lot for your talk. This is from Carlos Grelli. Regional pool of species is fundamental for advances in analysis. How can we define regional pool of species? Usually we use environmental niche modeling or species distribution models. Oh, sorry, my page closed here. I don't know what's happening. Just a second. Okay, I'm so sorry. Let me find here. Here, okay, sorry. How can we define a regional pool of species? Usually we use environmental niche modeling or species distribution models, but there is commission errors, especially because that are based on climatic variables. On the other hand, we can use the IUCN maps of range of species, but there is omission errors there. Do you have an idea of how we can advance in definition of regional pool of species?
Okay, so I think the most classic way of defining the species pool is not through SDMs. That is sort of a recent way of doing things. More classically, people used to delimit a biogeographic region and then assume that all the species that are within that biogeographic region are part of the species pool. That is kind of the most classic way of doing it. Then came SDMs, and people started defining the pool with respect to basically where a species can be found with respect to climate. I don't have a good answer for that. I know a while ago, Susan Harrison and what's his name, Cornell. I've seen some people trying to reflect on this because it is a very important question. Is it valid? Because it is a bit circular to go and do an SDM using the climatic variables to define the range of a species, and then use again the climatic or environmental variables to study the effects of metacommunities on the species pool or composition, etc. So there is some circularity in it. I don't think there is a good solution though. But I tend to think that if you can compare two different strategies, at least two different strategies, you can have a sense of whether the result you got was robust or not. So I would recommend using the IUCN, do the analysis, use the SDM, do the analysis, and then see if the results are the same. That's great. It means we are pretty certain that the result is correct. If it is not, then you would need to try to understand where does the difference come from. And that's also an interesting process that will take you a step forward. And I think it's useful to do it in that way.
Okay. The third question is from Professor Denise Rosa Feres. She thanked you. She said your talk was amazing. And she asked you if you see any way, even experimental or statistical, to understand the biological meaning of the spatially structured environment.
I think the best chances are trying to find situations like the one I showed in Chile, where you can separate a bit spatial structure from environmental structure. I think that's a good idea for it. Another approach, but I have not been fully successful with neither of this. I think it's it's a complicated issue. One strategy would be that. Another one would be to use a dispersal matrix that is independent of geographic distance. In a study with another student, with Charlotte Moritz, we had a dispersal matrix that was coming from an ocean currents model. And so we had a model of how the water moves, and the communities we were looking at disperse mostly through those currents. And so we expected that using that matrix to explain dispersal better than the environmental distance, or the geographic distance, could clarify the situation. And in fact, when we did the analysis, it did not clarify anything. On the contrary, it led us to more questions, because the dispersal matrix did not explain much, and the environmental distance ended up being highly correlated to space, but not to dispersal. So we had a whole level of new questions instead of resolving the issue. Another approach, experimental. Well, I think experimental could probably solve these kind of issues a lot better. And there is probably already something in there. published that I have not, I'm not on top of that literature. The problem with experimental is that, as I said at the beginning, it's usually done with very simplified communities, with species that have fast growth, etc. And so it's always an issue to try to think about, is it really general? Can you really generalize this into communities where species live a lot longer and they move across much larger spatial and temporal scales, etc. And then another possibility is to try to look at temporal patterns that have been very overlooked. It's likely that geographic distance, it remains the same throughout the years, but the environment doesn't, and the communities don't. So if we could somehow use that temporal variation, maybe we can try to tease apart better those components, but it's an open question.
Okay. The next question is from Juan Andrés Martinez Lanfranco. Do you have experience with more recent model-based approaches in contrast with algorithmic or distance-based approaches such as RLK to look at the fourth corner problem, such as CADS regression or joint species distribution models? Could those help us better extracting organizing rules for communities better than more traditional approaches?
I think it's definitely an area that deserves a lot of attention. That being said, I was very excited a few years ago when Mokani published a series of papers looking at the potential of using multiple species to model everything at the same time. We would understand individual distributions and turnover and species composition and their relationship to the environment. But then you start looking at the details, and well, that then developed into joint distribution models, and joint distribution models do have a lot of interesting aspects to them, but they are really useful to look at the conditional distribution of one species, condition on the composition of the surrounding community. But looking on the opposite side, looking at communities and what the different processes are, I don't know. I think there is a fine line between the models you use already have assumptions about the mechanisms that are important, and that includes joint distribution models, and that includes turnover models, and that even includes mechanistic models. But there is progress, obviously. I just think we are very far away from having the one solution, that the holy grail that will solve all these issues right now.
Okay, the next question from Adriana Carolina Acero Murcia. Could we expect a similarity in competition for trophic resources among species that are phylogenetically related? This given that species share similarities in the exploitation of trophic resources, and there is a homogenization of the habitat.
Okay. Can you repeat the part, similarities between? Can you repeat it?
If we can expect similarity in competition for trophic resources among species that are phylogenetically related.
Yes.
This given that species share similarities in the exploitation of trophic resources?
Yes. So if you look at the RD and Santerre paper that I cited there, or if you look at Viens or even the review paper I cited there, one of the tenets of using phylogenetic diversity in all of this is exactly that, is assuming that species that are closely related, phylogenetically related, are similar, and therefore they should be competing between them for resources, for space, for everything. And so, yes, the expectation is that phylogenetically close species will compete more strongly, and therefore they will be excluding each other at the local level. At the same time, you also expect those species to have similar environmental requirements. So there is again a scaling issue where you would expect that locally they exclude each other, but regionally they actually occur in the same places. But the basic assumption that is behind that is that the traits that have to do with that environment or that competition, that resource in the question, is conserved. And it could very well be that maybe the two species are very similar except when it comes to that resource. And so what I was saying in the presentation is that we need to consider whether or not the trait that is relevant for that particular competition or for that particular community is actually conserved before we can use phylogenetic diversity as a proxy for competition or environmental filtering.
Okay. And the last one from Luis Guilherme. He asked you, what do you think that are the main challenges for applying computer simulations to answer questions about mechanisms and processes that structure biological communities? Do you think there is a resistance in our field against this type of approach?
Oh, I think there are three different problems or barriers to this. One is from the simulation itself. Once you start simulating something, you can add all sorts of levels of complexity, and it's very easy to get lost in that. And the problem is that whatever simulation you end up doing has to be relevant for a real community, for a real system. And so it is tricky. It is tricky because you don't want to simulate something that is very simple, but doesn't resemble at all real communities. But you don't want to simulate something that is very complex, and it's so complex that you won't be able to understand it at the end. So you need a sort of a middle ground. In general, you want to start with the simplest because that's the way to understand what you're doing. But then you need to complexify it a little bit so that it contains the main elements of a real metacommunity. The second one is how to summarize and communicate the results of those studies. With the study we published with Tamara Monkemüller, in the study I showed, we struggled between ourselves and then with the reviewers on how do we translate something that took us a lot to understand. How do we translate it into a simple language without oversimplifying? Because again, if you oversimplify, then you don't transmit what the main elements of it were. So there is the communication aspect. And the third is, it's the interface with the reviewers and the journals and all that. there is, on one hand, a group of individuals who always think that simulations are great. No matter what you do, they are great. And then the opposite is also true. You will always end up with some reviewers saying, oh, no matter how complex your simulation is, it's too simple. The real world really doesn't resemble this. And so there is a little bit of chance on whether you end up with one or the other. There is a lot of communication that needs to be done in between. But mostly, I would say you need to understand yourself very well, the simulation system you generated, and go from the simplest to the more complex so that you understand what the different effects are. Because if you don't start with that basic understanding, then the rest, you know, the communication and the answering the reviewers and actually making your results relevant becomes impossible.
Okay, Christine, thank you very, very much for your talk, for your time to answer the questions. Thank you for everything.
Thank you for inviting me. It was great.
Thank you. And now I have some things to say here for you guys. Let me find it here. Okay. So I will say this in English before, and then I'll say it in Portuguese again. So we're sorry for everyone that had trouble with the YouTube link. We had some trouble here, but now everything is working. So we're going to share with you the link to the channel, okay? Not the specific links now. And again, so sorry. I would like to thank everyone for being here and people from the YouTube who had patience to wait for us to fix everything. Thank you very much. And now we have a cough break, and we can come back at 4 p.m. with the talk of Matthew Labeled. So don't lose it, okay? And I'll say this in Portuguese because I think this is more for who speaks Portuguese. Pessoal, lembrando que quem quiser escrever a pergunta em português para a gente, pode fazer a pergunta em português. A gente traduz aqui e passa para o palestrante, tá? Manda lá no Q&A, da mesma forma como vocês já estão fazendo, tá? E desculpa para o pessoal que está lá no YouTube, que a gente teve um probleminha aqui, mas já está tudo resolvido. Obrigada pela paciência de vocês, obrigada por todo mundo que está aqui. Vamos fazer então um intervalo. 4 horas a gente volta com a palestra do Dr. Matthew Labeled. Ok? Até daqui a pouco.